All right. Hello. Um, Welcome. My name is Robin McLam with Applied Natural Sciences, and I wanted to welcome each one of you and thank you for joining us for this webinar entitled Skeptical about Phytoremediation. Chris Gale is here with us and he'll be presenting today. He has 20 years of professional environmental consulting experience and he's implemented a wide variety of environmental site investigation, characterization, and remediation projects. For the past few years, he's been the vice president here at Applied Natural Sciences, where he's drawn from that expertise with a variety of remedial strategies when employing engineered phytoremediation technologies. And that's what he's going to talk more about today. So Chris will share for about 30-40 minutes, and then we'll have some time for questions. You should see probably at the top of your screen, not there, next to the little chat icon, there's also a question icon, and that's where you can submit questions at any point during the presentation, and they'll just come to me. I'll be compiling those for Chris and for him to address at the conclusion of the presentation. Also, I know some of you submitted questions beforehand, and Chris is going to try and answer as many of those as possible just in his presentation. But if you feel like you'd like to hear more, feel free to resubmit or adjust the question, and that's a good spot to drop it in there for me, and I'll be organizing all of those. So thank you again for joining us, and I'm just going to go ahead and turn it over to Chris now.
Great. Thanks, Robin. And thank you all for joining us. Looking at some of the folks that are online, I recognize some of the names we've worked with before. So I know there's some folks here that have some experience with phytoremediation. We're seeing that results in the poll that's up there as well. So what I'm going to go through is just kind of some general background, high-level background about phytoremediation, the applications, how it works, the mechanisms, and what are some limitations, perceived limitations of typical phytoremediation, and how what ANS does can overcome that. And then we'll go ahead and jump into a few case studies that kind of demonstrate the effectiveness to help address the skepticism why some folks are here. So we're going to go ahead and jump on in. So historically, phytoremediation applications typically used for soil impacts for contaminant degradation to actually break down the contaminants in shallow soil, also with mass removal to remove those from soil, stabilize it to make sure to limit the migration of contaminants within soil, and then also provide some revegetation in impacted areas. For groundwater, which is what's going to be mainly the focus of the talk here today, we do look at some hydraulic control as well as contaminant degradation and mass removal. Those are kind of the goals and applications for what we're looking for in a phytoremediation application. Some of the mechanisms by which phytoremediation works. There's a number of these mechanisms, and oftentimes there's multiple of these mechanisms working, and it's often dependent upon the constituent concern that you're looking for. So phytoextraction, really that means it's the uptake and the sequestration of contaminants in plants, and this is really applicable for soluble metals. So the metals will be removed from the subsurface and actually fixated in the plant. It's in the plant material itself, either in the root or further up in the plant. We can look at phytostabilization. So stabilization, this means that's basically the immobilization of those contaminants, either by absorption or fixation of that contaminant onto the root material, but also physically binding soils with the roots to help prevent erosion and leaching. Rhizofiltration is similar to phytostabilization, is where the COCs can be absorbed and precipitated on the plant roots. We also see rhizodegradation, which is kind of in situ degradation of soil microbes that's often mediated by root exudates. So we can actually get degradation of various compounds in the root zone itself. Phytodegradation, that refers to in-plant microbial mediated degradation of COCs. This is also largely endophyte assisted, so we do see especially with a lot of chlorinateds and other organics, you see actual degradation and you see metabolites within the plant material itself. And then phytovolatilization, so that's going to be when, for a lot of organic compounds, those that can be moved from the subsurface through the roots up in the liquid within in the plants themselves and volatilized through the stomata, where it can subsequently be phytooxidized or oxidized in UV oxidized in the atmosphere, and you can also get some as the water's moving up through the bark of a tree, you get some diffusion out into the atmosphere that way as well. And kind of a lot of what happens in one of the questions that was submitted in the registration, what is the fate of various different compounds within the trees or the plants? That's a complex question because it's often dependent upon what the nature of the compounds are. So a lot of the organic compounds, what we see is, and I think one of the questions was looking at leaf litter, and we typically do not see a substantial mass of the VOCs, the target VOCs that we're trying to remove in the leaf litter before the tree drops them. Typically, what happens is that as the leaves are getting ready to drop, the trees are pulling a lot of that moisture out of those, and the moisture is where most of that mass is still retained up in the leaves. Some applicable constituents of concern that we've looked at, a lot of agricultural inorganics, organics as well, solubilized metals. We've had lots of experience with various different volatile organics as well as semi-volatiles like 1,4-dioxane. I know this is probably the question on everybody's mind is what happens with PFAS. I think that's within the industry a very big question. We are starting to look at PFAS. There's some pretty good information out there that show that plants can uptake and sequester PFAS. There's some in situ degradation occurring of some of the precursor compounds, but I think the nature, the fate, and transport within the plant material, and we're still working on understanding that. There's a lot of different compounds. I think various different compounds behave differently within different species of trees. And so we are in the process of looking at that. We have a few projects where we are looking at PFAS fate and transport. So it's a little early for us to provide a whole lot of information on that at this time. So I know a lot of the limitations phytoremediation has been around since the early 90s. And I remember, in the late 90s and early 2000s, it was often served as kind of a silver bullet. And a lot of things it didn't work very well oftentimes because it was for a variety of different reasons. But there's some definite limitations to typical plants and trees in a shallow boring or near the surface, typically limited to shallow soil and groundwater, 10 to 15 feet. Basically, how far down can the roots get? If there's adverse groundwater or soil chemistry conditions, that can limit the effectiveness. We can also see that with limited geology. If you have shallow impermeable zones that prevent the root growth down to the impacted groundwater, that will limit your ability. Contamination, we often see that you will have a clean zone in the uppermost portion of the aquifer, but your contamination is deeper, and so the plants aren't able to get that because they're going to get the first available groundwater that they're going towards. And then also if you have areas of high rain, you're going to get shallow rooting, which would prevent and limit the amount of groundwater that the plants can actually take up. And then also high concentrations in certain areas. What we're looking at on this photo on the right, as you can see, kind of in the middle, the trees in the middle of this plantation are much, much smaller, and that's where the concentrations were much higher. So some of these high concentrations would typically limit the ability of trees or potentially even stress or kill trees if the concentrations are too high. Sorry, bear with me for a minute here. So what does our systems do? How the tree well system, what is it different? So the way that the tree well system works is that we really want to try and control where the plants are getting their water from, encourage the roots to grow deep towards the impacted groundwater that we're really trying to remediate. We can actually because of that, we can select specific depths. So we can actually eliminate or kind of block off that overlying, if there's a upper layer of clean groundwater, we can isolate that and have the trees go towards that underlying and pull water from that underlying zone. So we can actually target the depth at which we're trying to get the trees to consume water from. We have methods to access deep groundwater as well as fractured rock. The deep groundwater to date, we have gone as deep as 115 feet. That does necessitate that the groundwater be under a pressure head. So basically, it's going to have to be the potentiometric surface has to be about 35 to 40 feet. If it's deeper than that, then that becomes very difficult to get to. But we can get to deeper groundwater. We do have methods to do that, and we'll show that here in a second. We can also address, we've seen ability to address what many would normally call phytotoxic concentrations. Um, I think the way that ANS, the way that our tree well systems work, we actually creates a bioreactor effect that helps us address much higher concentrations than a typical phytoremediation plantation. We've planted in areas with Dean apple before, with 1,4-dioxane as high as 2,000, almost 2,000 milligrams per liter without seeing phytotoxic effects in the trees. We've integrated our systems with a variety of other remedial technologies to really capitalize on some synergies and also create combined remedy strains that are achieve clients' goals most efficiently as possible. And then our systems also can help eliminate or really limit the need for supplemental water after the trees really get established because they're consuming groundwater, there's no need for additional shallow irrigation. So how does the tree well technology work? So the tree really acts as a solar pump in our systems. So here's a cross-section schematic of one of our standard tree well sections. The green here is what we call our root sleeve liner. So we'll drill down with a large diameter auger down towards our target depth. So in this case, we're targeting something that's much further below groundwater. As long as the soil is cohesive enough to stay open, we can drill down, put the liner in, and then backfill with the sandy loam soil. Typically, that's amended with about a 10%, 5 to 10% organic matter backfill, and then we can put piezometers in here if we want to monitor groundwater coming into the system. But in this case, what the tree is doing is pulling groundwater in from that lower zone up through that soil column. And because we do have a fairly large soil column, we're typically drilling with a 42-inch diameter auger, and we'll drill well into groundwater so that provides sufficient mass and organic matter. So we do see a lot of in situ degradation, especially of organic contaminants. And we can see that as the groundwater is migrating upwards through that soil column. And then in addition, we also see the rise of degradation. So with degradation within the root zone, that's mediated by the tree exudates, and then further degradation within the tree itself. And then as those residual concentrations continue to be transported through the tree, we do see some photovolatilization and the UV degradation ultimately. So we have a couple options. So the first one we're kind of looking at on the left here is our standard tree well unit. It's an open bottom unit. We use these for shallower groundwater, or even we can get to those underlying zones. But it's really kind of looking at hydraulic control and kind of a bioreactive barrier. So we are relying also on the in situ degradation that we're seeing within the tree well unit itself, that bioreactor effect. And then on the right is kind of what we call our straw well system. And this is how we can actually reach to that deeper groundwater zones and reach into fractured rock units as well. So you're seeing this is a closed bottom unit. So we'll drill down to a few, probably 5, 6, 7 feet below the potentiometric surface, below where our targeted groundwater zone is or where groundwater will be encountered, to provide enough room so we'll get some additional degradation and provide us that soil column that we need. And then place a bentonite seal at the bottom, put the liner in, the closed bottom liner, another bentonite seal in the bottom of that liner. And then we can come back and drill through that, usually with a rotosonic drill rig, and we'll put a dual screened well in. So the lower screen is in the target zone that we want to get into. So like I said, we've gone as deep as 115 feet before. And then the upper screen is really set so it's right above the top of that inner bentonite seal. So as the tree consumes groundwater, because it's kind of a closed system, semi-closed system, where the only place that is open is through that straw well. So as the tree consumes water, it draws down the water level within the unit itself, and then the potentiometric head pushes and drives additional water in through that straw well up into the system to replenish the volume, the water in that system. And then the degradation, same degradation processes continue to occur in that tree well, in the straw tree well unit itself. Another system that we've just recently kind of developed is a tree well system with a replaceable cartridge. So this is using that same straw tree well system where the treater is using groundwater in the closed bottom system. And then hydraulic head is what's used to replenish the water in the system. In this case, what we're using inside that straw unit itself, we can put in a cartridge. A cartridge can be filled with a treatment amendment to either treat certain agents, like high concentrations of VOCs, pesticides, oxidized materials that could potentially be phytotoxic, or sorb certain compounds such as metals or PFAS potentially. And this would be a replaceable cartridge that once the sorptive or the treatment capacity of that media is reached, we can actually remove the cartridge and replace the media that we're looking for. And it's really kind of and then not trying to get full treatment, but knock the concentrations down, and then we get further treatment within the tree well system itself. So I'm going to jump now into some of our project summaries, kind of some examples that we're looking at. One that we installed in Louisiana in 2015, one in the EU in 2013, Pennsylvania in 2012, and Florida in 2013. So these are all fairly mature installations that we have quite a bit of data on. So the site in Louisiana, it's primarily, it's a mixed VOC site and very high concentrations. Some of these areas had kind of DNAPL type concentrations. So this is how we kind of manage the phytotoxic concentrations. And then also this is where we started looking at how do we address and reach those deeper groundwater zones. So as I said, this site in Louisiana, including DNAPL, so our guys, some of the installation had to be in full level B or level C. And then because it was in Louisiana, the photo on the right, we're drilling on pretty swampy conditions and installing on swampy conditions. So everything was done on rig mats. And this was an agriculture facility. Sorry, no, it was not. This was a waste oil facility. A few different treatment options were looked at here. So for the shallow zone, the first three on the left, we're looking at kind of a standard tree well unit where just the backfill was amended with organic matter, so the 5 to 10% organic matter in an open bottom tree well system, kind of targeting that sand, that lower sand zone, or upper sand zone, sorry. And then we also looked at another scenario where we added some zero valent iron to try and help reduce concentrations. So that ZVI was really added at the bottom of the unit. And so as groundwater would flow upwards through the bottom, through that ZVI, kind of trying to reduce concentrations to mitigate potential phytotoxic effects. And then a third scenario was looking at the addition of both the ZVI and Deramind. Deramind is another product that induces that'll help with reduction of concentrations. And then we also looked again here at two different straw units to look into getting into the lower sands. There's one about 75 feet, and then this is where we got down to about 115 feet. So what we're looking at here, this is kind of looking at concentrations. So there's a paired piezometers that are installed within the tree well units themselves. So looking at groundwater that's coming in from the bottom of the unit here, and so that's what we're seeing in the closed symbols, the red and yellow symbols that with the closed symbols there, and then the open symbols are looking at what's at groundwater concentrations closer to the top of the potentiometric surface, so just below the root zone. And what we actually saw here is we see multiple orders of magnitude. So the concentration on the left is total VOCs in milligrams per liter. So this is total VOCs, and you can see 1 to 2 to 3 orders of magnitude difference just in the vertical difference as it was migrating upwards through that soil column. So you do see a lot of degradation happening before it even hits the root system of the tree. And comparing this, you see a similar trend here looking. So what we're seeing is groundwater concentrations, again, At the top in the purple is groundwater coming into the system, and then flowing through the ZVI, the Daraman, and ultimately into the sandy loam soil, the standard organic amended backfill soil. And what we see is similar behavior and similar order of magnitude reduction. So as it's flowing up, and I think that the big picture for me is that we see a similar reduction, similar order of magnitude reduction with just the backfill soil. That while the ZVI and the Daraman do provide some extra degradation, it's not substantially more than just the backfill soil, the organic amended backfill soil itself. So this is kind of showing a detailed of how we got down to the 115 feet. So again, this is using hydraulic head to access that deeper water as the tree, again, consumes that water head, hydraulic head, replenishes in the unit, the concentrations. So a similar trend as we're seeing here. So the groundwater entering into that through that straw well, higher concentrations and a multiple order of magnitude reduction by the time it got to the top of the groundwater surface as it's migrating upwards through that tree well column. And we've used these same we can also use these tree wells again to the straw tree wells to reach fractured rock zones as long as there's, again, the groundwater can come up sufficiently within the tree well unit, as long as it's under enough hydraulic head. Our next case study is a site that we did in the EU. It was a 1,4-dioxane site. Really kind of planted initially to provide hydraulic control, and the thought on this one was that the goal of it was really to basically have the trees capture the groundwater and transpire and have the 1,4-dioxane ultimately degraded within the atmosphere. So it's a 1,4-dioxane plume with a source area with more than 100 milligrams per liter. I think some areas where we had stuff close to almost 2,000 milligrams per liter. Groundwater impacts in two separate water bearing zones. One is about 4 to 6 meters and one about 7 to 10 meters. So former is a large chemical production facility that used to actually produce 1,4-dioxane. Dioxane concentrations were, like I said, up to 1,500, migrating towards a nearby surface water. So a couple of different zones that, like I mentioned, the groundwater, we're really from the source area migrating towards the property boundary and a nearby surface water area in a couple of different zones. So we had to try and target the shallow zone as well as that deeper zone here. And typical containment alternatives for this would be really labor and energy intensive and not really attractive from an economic or sustainability standpoint. So that's why they chose the more nature-based remedy for this site. So the remedy on this was 250 different straw tree well units, 185 in that shallower zone and an additional 54 in that 7 to 10 meter zone. The system is designed to account for groundwater flow across the planting area and to ensure that we had a sufficient number of trees to capture the volume of groundwater that was flowing through that zone. The straw tree wells allowed access to the deeper water. The closed bottom units constructed with a dual screen, again, to allow that groundwater to come up and into the system. Here's another just cross section showing kind of the root sleeve, the clay. This is our straw unit. So as we're seeing, those are set at two different depths, 4 to 6 meters and also 7 to 10 meters below ground surface. So this is a plot from one of the wells, and it's just kind of, there's a lot here to unpack. But the blue line, the solid blue line, is the concentration in a nearby monitor well, and you see a steady decrease following the installation. The dashed lines, the dotted lines, are the concentrations within various different tree well units. And you see they do fluctuate quite a bit, and those fluctuate seasonally. So what we were seeing is that as the tree wells, the concentrations would increase as the trees are actively pumping and drawing that mass towards them. This site also looked at because as I mentioned, they were primarily looking at volatilization, phytovolatilization. So looking at trying to see how much of the mass was being transpired directly to the atmosphere, and did this with basically putting some Tedlar bags around leaves, and then trying to record the mass that was coming out, that was being actually fluxed into the atmosphere. And turns out over, I think, three or four different monitoring events doing this, the estimated mass flux was less than 0.1% of what was expected. And that's based on the consumption rates of the trees as well as the concentrations of that flowing into that zone. Some work done by others, not us, did identify some dioxane monooxygenase and aldehyde dehydrogenase. So, and a number of enzymes that were associated with that biodegradation of 1,4-dioxane. I think there's been quite a bit of research on this, and may even be some folks on this call. I know some folks that had signed up for the call that are probably more familiar with this additional work. But functionally, what we see is that we are seeing biodegradation, substantial biodegradation happening within the tree well systems themselves. So I think it was substantial degradation before it even hit the trees themselves. So what we see, this was actually the dioxane plume in a shallow aquifer within 1 year of planting. As the trees after one growing season, you can see the trees are actually pulling the plume towards the plantation itself, and it would shift back and forth during the winter season, and then the active summer growing season. The shape of the plume actually goes more towards the trees when the trees were actively transpiring and consuming groundwater. What we saw was a steady increase in growth through 2020. I think that this site has just recently the trees have been removed because I think they're going to reuse that portion of the site for a new facility. But as the trees continue to grow, and the canopy increases, you continue to increase your groundwater consumption rates, which in turn increases the functionality of that system. So kind of key results and take-homes on this, did provide full hydraulic containment after the first growing season. We do see a pretty high mass uptake of 50 to 100 kilograms per year. The evapotranspiration accounted for less than 0.1% of the 1,4-dioxane mass that was being removed from the system. Most of that mass appears to have been removed from biodegradation of the 1,4-dioxane within the tree well soil column itself. And there were no evidence of phytotoxic effects due to 1,4-dioxane concentrations, like I said, 2,000 milligrams per liter. Next site is in Western Pennsylvania. It's a chlorinated solvent site. We're really looking to reduce contaminant mass in groundwater and provide some hydraulic control. This is a fairly complex geology site. So in the early 2000s, they identified chlorinated VOCs, predominantly TC and some degradation products, along with chloroform and TCA, in surface water and sediment near one of the two outfalls leading from the plant wastewater treatment facility. This is a former electronics manufacturing facility making commercial and household appliances. So let's see, jump to the next slide here. So the original strategy for this was to address the source area mass using SVE, in situ chemical reduction, and in situ bioremediation, and then also help provide hydraulic plume control further of the down gradient groundwater and to mitigate offsite migration. And the tree well phytoremediation system in 2012-2013 is this initial treatment system pilot study that included 52 units that were installed in 2012-2013, and then subsequently further expanded in 2016-2017, further downgrading. So 52 tree well units basically looked at 25 of those units were in the shallow soil, so about 5 meters or 15 feet below grade. And then 27 of those were installed in the upper fractured rock down to about 30 feet. This was planted with a combination of hybrid poplars and willows. And this was really conducted to look at the efficacy of the tree well technology to see if it could remove the dissolved phase COCs and prevent that offsite migration. So what we're looking at is from the planting through 8 years after planting, you see pretty substantial degradation and steady degradation in one of the monitoring wells within that pilot study area. Again, we're looking at total VOCs, total chlorinated VOCs on the left here over time, and you see again a steady reduction in concentrations. Here's a photo of that pilot study, the plantation after 11 years. So you can see very substantial, robust growth, big, beautiful trees. Feel these look much nicer rather than a fenced-in treatment compound with stacks and other various equipment. So based on the success of that initial pilot, the additional 166 tree well units installed further down gradient. So in the tree on this, in the background photo here, you can see the tractor trailers, the semi trailers there near the top, and then the original planting area is near the center and top of the photo. And then these units were installed further down gradient in 2016 and 2017, just after our planting. This is another photo that was taken I think it was taken last spring. So shows and this is in the down gradient area. So you can see the growth over a few years. The tree well system performance. So this is in that on a down gradient area. We see again steady decrease in concentrations. No concentrations again, total VOCs in milligrams per liter on a log scale on the left. So we see a steady reduction in concentrations at this site as well. Another last site I believe here is our one we had near Sarasota, Florida. It's a 1,4-dioxane site. We're looking to reduce contaminant mass and provide some hydraulic control. So this is a 2.5 acre site that had mature full vegetative canopy, as you can see on the left side of the site. On this photo on the right, left and lower portion of the facility is fully vegetated. The problem is that there's some remaining source area. There was an existing pump and treat system working at the site that wasn't really working. The 1,4-dioxane plume was migrating off-site to the west, to the left on the picture, in a bedrock aquifer. And there are two different aquifer media here, the shallow, which is 0 to 2 meters, 0 to 6 feet. So groundwater is very shallow, but that was very sandy, and that was largely clean. That could have been largely because the trees that were existing were rooted into that shallow system itself. But that underlying fractured bedrock aquifer still is where the 1,4-dioxane mass was really migrating. So here's a couple of photos. The big photo on the right shows the plantation shortly after the installation with the mounds and our tree well system, and then the other two photos on the left here show some of the trees as they're continuing to mature. So initial groundwater flow at the site in March 2013 was towards the west-northwest. And then we did include some pressure transducers in 14 of the units to kind of monitor the groundwater drawdown in these areas. So the 154 unit system did provided some plume control. These are planted with a combination of slash pine, willow, sycamore, cypress, and laurel oak. So trying to evaluate the effectiveness of this system, the consultant on this did run through a groundwater extraction model or bait and transport model, and the model did predict that the system provide capture, assuming that each of the trees was pumping at a rate of 20 gallons per day per tree. So that predicted groundwater capture at 20 GPD per tree. So in 2014, groundwater conditions demonstrated capture, and this is based on the potentiometric surface from groundwater monitoring wells, as well as the piezometers and pressure transducers within the system itself. Concentrations of 1,4-dioxane in the planting area and on the upper left plot show that almost immediately following installation, um, substantial decrease in 1,4-dioxane concentrations, and even seeing concentrations being reduced in the source area as well as down gradient outside of the planting area. And it's a fairly quick response following the planting. So takeaways from this Sarasota site is that they were able to shut the pump and treat system off once the tree well system was established, and it saved about $300,000 a year in O&M costs. Able to demonstrate that the active remediation of the tree well system was providing hydraulic control with a much lower O&M cost. Key point here, and the phytoremediation system is not a no O&M system. It is a low O&M system. It's still an active system that does require some maintenance and monitoring, but it's much less than an active pump and treat system or other active mechanical system. It was anticipated that the plume would reach cleanup goals within 5 to 7 years. However, no further action was received 4 years after the installation and planting in December of 2016. So I think that addresses what we have for now. I think we can jump into any questions, Robin.
All right. Yeah, feel free to keep, if anyone wants to add any questions over in the chat. But a couple things, Chris. So actually looking to see if you could talk a little bit more. I think the theme would be a little bit more about monitoring and maintenance. So, like evaluating water consumption in trees, accounting for dormancy in winter, and just sort of how are the systems monitored as it goes on.
I guess maybe first we'll talk about how we account for dormancy in the wintertime, because that's more, we engage with that kind of early on in the design phase. And so we need to know and want to know what the groundwater flow rates are, what the groundwater velocity is through that system. And we try, because if the objective is plume control, we need to make sure that groundwater that's entering the system during the dormant season does not flow far enough and get out of the treatment system before the trees come out of dormancy. So we will try and design that system such that there's plenty of buffer along the groundwater flow path so that we have trees installed so that groundwater will not flow past the system before the trees come out of dormancy. In terms of monitoring and establishing groundwater consumption rates, it's easier to do with the straw tree well systems because those are closed systems. There's a couple ways we can do it in the straw system. You can essentially put a packer in the straw below the upper screen to isolate the groundwater from coming back in and create a true closed system. And then you put in a pressure transducer and you can monitor the drawdown within the unit over a couple of days and get a rough approximation just based on the volume of that unit and understand how much water the trees are consuming approximately. Another way to do it is sap flow monitoring. That's something Paul, our colleague here at ANS, has quite a bit of experience with. And generally, at mature trees, so the groundwater consumption rate is highly variable on a number of factors. Primarily, it's the amount of leaf area, so how much can the leaves transpire per day. So it relies on direct sunlight, relative humidity, temperature, wind. There's a lot of things that drive into it, so there's a lot of variables. But kind of for those that are out there wondering, a ballpark estimate for a mature tree is about 1 to 2 liters per day per square meter of leaf area that's available in the tree. In terms of monitoring for tree system, like tree health, like we need to continue to monitor. These are nature-based systems. They're natural systems. There are natural pests, natural stressors that can affect these systems. So routine inspections for pest infestations, beetle infestations, or are there fungal infestations, things that can be addressed with the application of insecticides or fungicides. and as long as you can get them early enough, we can, you can treat those and address those problems. But the trees do need to be monitored for those types of stressors. In terms of groundwater monitoring, depend what we're looking at. We try to keep the pressure transducers are a great way to demonstrate active groundwater consumption because you can actually see the drawdown relative to wells outside of the system. Within the system, you can create your pangeometric system, but we can also actually see sometimes, it depends on your groundwater permeability, but what we can actually see sometimes is you can get groundwater mounting because the trees are pumping groundwater into that system. But the pressure transducer data can show kind of diurnal pumping effects. So you can see when trees are actively transpiring versus dormant day and night as well.
Okay, thanks, Chris. So another question someone submitted, and we get this often. Could you talk a little bit more about tree selection? So you mentioned a couple different tree species in your talk, but how do we decide that? Are there specific trees we tend to go with? Do we use native trees?
Yep. So in within the industry, hybrid poplars and willows are two of the most frequently used because because they root deeply, because they use a lot of water. We do try and include a variety of species in most of our plantations because in the event that if you planted an entire installation with a single species, and then there was an infestation, a pest that targeted that species, you would lose the entire planting. Whereas if we get a variety of species, we can it's a little less susceptible to infestation and losing the entire planting. Um, we do sometimes try to use native species, but we will definitely use species that are going to be suitable for the climate at the site. Um, but we do need to use trees that are capable of rooting deeply. Trees that we do want to use trees that grow quickly and use a lot of water.
And this is another, I know you covered some of this, but do you want to talk a little bit more about any geologic or hydrogeologic design considerations? If they're, I don't know, some non-starters or particular project requirements you're looking at?
Yeah, so we do have limitations. Basically, if your groundwater flow velocity is too high, the trees just can't keep up with it, or it becomes impractical. We would need to plant multiple acres. So there are some limitations. High permeability zones we can treat as long as the gradient's pretty flat and there's not a lot of water moving through those systems. But that's a lot of the information that we look for and we talk with on the front end, and there's plenty of sites where we say we just can't address that because I would need 10 acres of trees. It's just not practical. Other limitations, like I think I briefly touched on it, is there we can kind of work through it if there's a lot of clay layers, and that's kind of what we designed for. Is that if there's interbedded silts and clays within your target aquifer zone, that could limit that upward migration from our target zone or get really stratified within there. We can help break that up during the installation with a large diameter auger. Typically what we'll do, like I said, is we'll drill with a 42-inch auger, but we can then transition down to a smaller auger, a 24-inch or something. And we'll drill into our target zone. You may drill 5, 6, 7 feet into that zone and and that's really just trying to homogenize and break up any of those interbedded low permeability layers. And then also while doing that during the installation, we can add some of our amendment or our potting soil, basically the organic amended soil, and try and reverse auger that or drill it into that formation so we can break those impermeable layers up and provide a better conduit for the trees to pull that water out.
Great. Thanks, Chris. I think that covers most of the questions that we've gotten. If you have any last minute questions, you can throw them in now, but I think, and we're getting close to the time too. So thank you all so much for joining us. And Chris, thank you. for sharing all of that great information with us. If people want to follow up, what would be, if they have specific questions? I know some people look like they had sites in mind. What would be a good way to follow up?
Please email me or give me a call if you have any further questions or want to chat further or more specifics.
All right. Well, thank you all again, and we'll be able to, I believe, share this video as well. So you might receive one more email from us in conclusion of this. So thank you so much. And Chris, could you put your number and email maybe back on the screen?
Yeah,
That'll
let me
be
get to that.
possible.
Let's see.
Um,
Just go to back to the intro slide here. Sorry.
yeah, back to the intro slide, and of course you can email me as well and put people in touch with Chris too.
So here's my contact information.
There
If
you go.
anybody has additional questions or sites in particular that they'd like to discuss, I'm happy to chat.
All right. Well, thank you all very much.
All right. Thank you very much, everybody.